Hello, everyone, and welcome to another episode of Emerge. Thank you very much for joining us. My name is Stoyan Stoichev, and I'm from the Resin Bio team. Today, I have the pleasure of hosting Patricia Bortel. I'll give you a little bit more information about her just in a minute. Just to start off with the usual announcements that we are recording the session, and we're going to post this on our YouTube channel, the Resin Bio YouTube channel. Probably by end of this week, definitely early next week. So you can review this recording. Please post your questions. We have plenty of time to tackle those. Hopefully have a nice discussion. It's a really nice presentation today, talking about technical aspects of phosphoproteome profiling. So yes, I'm really happy to be able to host Patricia from University of Vienna in Austria. She's currently completing a PhD. The Institute of Analytical Chemistry, and she mainly focuses on optimizing affinity-based enrichment strategies. So today she's going to talk about some of those optimizations that she has performed and really highlight some of the more important metrics to keep in mind when we're trying to get the most of our phosphopeptide enrichment pipeline. So Patricia, thank you very much for joining us. Thank you for the time, and I look forward to your presentation.
Hi, thank you for the nice introduction. So I'll just go ahead and share my presentation. Yes, so I hope you can see my screen.
Yeah, perfect.
Great. Okay, then yeah, thank you for the introduction and the invitation to the webinar. So today I would like to present to you a collaborational project which was established during an internship I did at the Olsen lab. I'm originally from the Gerne lab at the University of Vienna. And in our work, we focused on the systematic optimization of automated phosphopeptide enrichment for high sensitivity phosphoproteomics. So as probably a lot of you know, protein phosphorylation is a highly dynamic post-translational modification, and it plays a very important role in cellular signaling. And protein phosphorylation is an quite popular study object of the proteomics community, with the possibility to analyze up to thousands protein phosphorylation sites across different cellular contexts. However, there's still ongoing research trying to identify the functionality of most of these phosphorylation sites. And mass spectrometry has actually proved itself to be one of the best platforms for large-scale and unbiased identification and quantification of such phosphorylation sites. However, due to the low abundance and relatively low stoichiometric nature of phosphopeptides, there is still a need to enrich for these species for a deep phosphoproteome before the LC-MS analysis. And the most popular materials that are being used during phosphopeptide enrichment are the IMEC and the MOIC materials. So these are the immobilized metal affinity chromatography or the metal oxide affinity chromatography. And both of them usually rely on transition metals such as iron, IMEC, or iron, zirconium, or titanium. And regardless of the material, the effectiveness of these phosphopeptide enrichment strategies largely depends on the parameters during the enrichment. So for example, what can play a role is the peptide to beads ratio, the composition of all buffers used during the workflow, such as the loading buffer, the wash buffer, and the elution buffer. For example, the loading buffer can contain some kind of competitive non-phosphopeptide binder, such as glycolic acid, or high percentages of trifluoroacetic acid to avoid the binding of highly acidic non-phosphorylated peptides. And also the binding time, so the time for how long you are actually incubating your sample with beads, can play an important role on the resulting phosphoproteome. And in the last years, significant improvements have been made in the phosphoproteomics field, mainly by new highly sensitive mass spectrometers, as well as the implementation of data-independent acquisition, and also software tools that are capable of analyzing DIA data without the need of spectral libraries. And in this regard, these rather technological achievements have a bit overshadowed the improvements that can be made by adjusting the parameters of your actual phosphopeptide enrichment. So our goal in this work was to evaluate key experimental parameters and optimize them in our automated phosphopeptide enrichment workflow with a focus on low input samples. So our workflow implements the zirconium IMEC HP beads from Magresin on the Kingfisher platform. And as an input for the workflow, we used 30 micrograms of peptide from a triptych digest from A549 cells. And we used this peptide amount for all of our standard evaluation because this represents the optimal amount for subsequent LC-MS analysis with the whisper gradients on the EvoStep1 and subsequent MS analysis on the Explorer 480. And as a software, we use Spectronaut in library free mode. So here you can see an overview of the phosphopeptide enrichment workflow itself. So you basically start by choosing your sample with your desired peptide input amount. You mix your sample with your loading buffer, and afterwards you incubate this mixture with your beads. And after several washing steps, you elute the phosphopeptides from the beads with an elution buffer. And within this workflow, we wanted to evaluate the peptide input in cells, the sample volume, so the concentration of the sample. Then we wanted to check whether the loading buffer volume, as well as the concentration of glycolic acid, plays a role. And then in the enrichment process itself, we wanted to evaluate the binding time as well as the bead volume. And in the very last step of the protocol, we also evaluated the percentage of ammonium hydroxide in the elution buffer. So the first parameter that people might think about adjusting when they don't see the desired results in their phosphoproteomic workflows is the peptide input amount. So here we tested 5, 15, and 30 micrograms of peptide input. And as expected, we saw that with an increasing peptide input amount, we also got an increasing number of phosphopeptide identifications. And important for the rest of the talk here is that when I'm talking about identified phosphopeptides, I always mean those with a localization probability of above 0.75. And we also observed that the localization probability, as depicted here in the violin plots, scales with the peptide input amount, which makes sense because if you have a rather small peptide input amount, the peptides you enrich, so the phosphopeptides you enrich, will also be of rather low abundance. And when you use these peptides as an input for your LC-MS analysis, the chance that you're producing poor quality MS2 spectra is higher, and that complicates the subsequent localization of the phosphosite on the phosphopeptide. And the last observation we made with this initial evaluation was that phosphosite populations from the different input amounts highly overlapped, as you can see in the Venn diagram here, with the more abundant phosphopeptides being enriched in all species, while the rather low abundant phosphopeptide species were preferably enriched only with the higher input amount. So then we also wanted to check the sample to loading buffer volume ratio. And we did so by first diluting the sample in increasing volumes while keeping the loading buffer volume constant. And in the second step, we kept the sample in a constant volume and increased the volume of the loading buffer. So we saw that when we diluted the samples with increasing volumes, we actually got decreasing phosphopeptide identifications, whereas we observed that when increasing the loading buffer volume, this didn't have such a significant effect on the phosphopeptide identifications. And interestingly, we saw that when decreasing the sample volume, the enrichment efficiency got a bit worse, which could be explained by the fact that when you use higher volumes of sample, you actually also dilute your loading buffer. So it might be that the ratios of some chemicals in the buffers aren't optimal then anymore. Then during the enrichment process itself, we first evaluated the bead volume. So it is generally known that you shouldn't use a too high bead volume because that can lead to a lot of unspecific binding. And that was what we also observed. So we saw that with our standard input amount of 30 micrograms of peptides, when we used 5 microliter of the beads, we got the highest phosphopeptide identification numbers. When we increased the bead volume even further, we saw that the phosphopeptide IDs started to decrease again. And in contrast, the non-phosphorylated peptides started to get enriched. And when we decreased the bead volume, the phosphopeptide IDs dropped, but also the enrichment efficiency, so the ratio of the peptides to the phosphopeptides, got better. We also had a look at the multiplicity of the enriched phosphopeptides, and we saw that when there were rather low bead volumes available, that we enriched more of the multiply phosphorylated phosphopeptide species. And we also observed that when using low bead volumes, the median phosphopeptide PI of the enriched phosphopeptides was rather acidic. So the enrichment was rather more specific with the lower bead volumes. So during the enrichment process itself, we also evaluated the bead binding time. And here we checked a binding time ranging from 5 to 30 minutes. And we saw that there wasn't a huge effect, but still we got the most phosphopeptide IDs with 20 minutes of binding time. And also on the multiplicity here, we didn't see a significant effect. So then we also wanted to have a look at the glycolic acid in the loading buffer. So as I explained in the introduction, glycolic acid can be used as a competitive non-phosphopeptide binder. And in our experiment, we definitely saw that it has this function. So when we tested increasing molarities of glycolic acid in the loading buffer, we observed that the phosphopeptide IDs started to drop, but the enrichment efficiency, so the ratio of the non-phosphorylated to the phosphorylated peptides, got better, which means that in very high concentrations, glycolic acid does not only compete with non-phosphorylated peptides, but also with phosphorylated peptides. And interestingly, we also saw that with increasing molarities of glycolic acid, we enriched a higher number of multiply phosphorylated species, which means that under very competitive conditions, the multiply phosphorylated species will preferably bind to the bead. And again, we also observed that with increasing molarities of glycolic acid, the enrichment got rather more specific with more acidic phosphopeptide PIs with the higher concentrations here. And the last parameter that we evaluated in the very last step of this workflow was the percentage of ammonium hydroxide in the elution buffer. And also for this parameter, we didn't see a significant difference. We tested here 0.1 to 2%, and the best percentage were either 0.5 or 1% in terms of the phosphopeptide IDs. And also for the multiplicity, we didn't see significant differences here, although the multiply phosphorylated species were a little bit lower with the very low percentages of ammonium hydroxide. So to sum up our findings, we can say that mainly the sample volume, the sample input, as well as the proportion of glycolic acid in the loading buffer and the volume of the beads play a significant role on the enriched phosphopeptide species, whereas the binding time as well as the percentage of ammonium hydroxide and the loading buffer volume do not play such a significant role in the enrichment process. So when comparing the different parameters here, it becomes evident that there is no one solution fits it all. So the users should rather choose which metric is most important for them and then adapt the parameters to fit their specific needs. So here you can see that when you want to go for very deep phosphoproteomes and get the most phosphopeptide IDs out of your sample as possible, you should go for 5 microliter of beads in combination with 20 minutes of binding time. Then you can use either 0 or 0.1 molar glycolic acid, as well as 0.5% of ammonium hydroxide. But maybe when you observe that you have a lot of unspecific binding in your workflow, you should try to use rather competitive binding conditions. So if you want to go for a higher enrichment efficiency, you could use a lower bead volume, such as 1 microliter, with a decreased binding time of 5 minutes, a high molarity of glycolic acid, and 0.5% of ammonium hydroxide. And this is also valid when wanting to go for the rather multiply phosphorylated peptide species. So here you should also go for the rather competitive binding conditions with low bead volumes and high percentages of glycolic acid. So in this very first evaluation of our workflow, we thought that the parameters that you employ can significantly improve the resulting phosphoproteomes. And another way to improve the phosphoproteome depth, which has been suggested before by the community, was to perform a so-called sequential enrichment. And the easiest way to do so is to just use the flow through from the previous enrichment and just restart all of your incubation, washing, and elution steps. And the first thing we wanted to test was whether we can reuse the beads from the first round for second enrichment, or if it would be better to use a new aliquot of beads. And we tested this with a 2-round sequential approach. And as depicted in the bar plot here, when we focus on new beads in round 2 and reused beads in round 2, we actually observed that reusing the beads gave a bit higher phosphopeptide identifications in the second round, and it also slightly increased the enrichment efficiency. So therefore, we stuck actually with reusing the beads from round 1 to get a higher phosphopeptide recovery in round 2. So the next thing regarding sequential enrichment we wanted to test was how many rounds of sequential enrichment are actually needed to efficiently deplete a peptide input sample for phosphopeptides. And we tested this quite extensively by performing 6 rounds of sequential enrichment with 4 different peptide input amounts, which were 2.5, 5, 10, and 20 micrograms. And importantly here, we collected each fraction of each round separately. So after each round of sequential enrichment, we retrieved the alloy and replaced it with a new elution buffer. And subsequently, we also measured each of these fractions separately. We observed that with all peptide input amounts, that with subsequent rounds of sequential enrichment, we enriched less and less phosphopeptides. But what was quite interesting to see was that with the highest peptide input amount, that was 20 micrograms, we still enriched up to 2,000 phosphopeptides in the very last round. And regarding the enrichment efficiency, we saw that enrichment efficiency dropped for all input amounts with increasing rounds, but this drop was more evident for the lower peptide input amounts. And although it is quite nice to see that in round 6 we can still enrich a lot of phosphopeptides, it of course matters which kind of phosphopeptides these are, because if those enriched in round 6 were already enriched in round 1, we won't gain a lot from it. So we calculated the cumulative unique phosphosites. So to round 1, we added with each subsequent round only those that haven't been enriched in any other round before. And what we saw was that for the lower peptide input amounts, the highest increase in cumulative phosphosites came from the second round of enrichment, whereas for the higher input amounts, it was still beneficial to do a third round of sequential enrichment. And we also had a look at the abundance of the phosphopeptide. So here you can see in each column one replicate. And we observed that the phosphopeptide species that were enriched in more than one round were those that were rather high abundant, which explains why they are still present in the rather late rounds of the enrichment. And however, although it is nice to get more phosphopeptide identifications by such a sequential enrichment, one has to keep in mind that sequential enrichment does not only involve more sample preparation time and subsequent MS measurement time, but there is also no standardized approach on how to handle multiple enrichments from a quantitative perspective when we consider that we have a high redundancy of phosphopeptides that were identified across all these sequentially enriched fractions. So that was the question that we asked ourselves. And a potential solution that we came up with was to pool the alloys. So here in this experiment, we tested the so-called pooled approach, where we reused the same elution buffer for all sequential enrichments, and the so-called cumulative approach. So in the cumulative approach, as I told you before, we collected each alloy from each round as a separate fraction and measured it, and then cumulated the total phosphopeptides afterwards. Whereas for the pooled, we only had one fraction to measure. We tested this approach for the low input amounts, which were 2.5 and 5 micrograms of peptide. And we observed a gain of approximately 7% when combining the first and the second enrichment for 5 micrograms of peptide. However, we didn't observe such a gain in the 2.5 micrograms. And the boost that we can see here in the IDs could be as depicted in the box plot here, due to a slight boost of the phosphopeptide intensities in the pooled samples here. And we also explored this type of pooling approach from a quantitative perspective. So here we calculated the CVs of the pooled fractions and compared it to the cumulative approach as well as to the single rounds. And reassuringly, we saw that the CVs were not significantly altered by this pooling approach and remained with a median of below 20%. And then we tried to exploit the benefit of maybe enriching different phosphopeptide species in each round of sequential enrichment. So here we applied the observation that we made in the early evaluations of this project, in which we saw that when using rather low bead volumes, we enriched rather multiply phosphorylated species. So we performed here a sequential enrichment in which we used 1 microliter of beads for the first enrichment round. Then for the second sequential enrichment round, we added another microliter of beads. And for the third round, we added another 2 microliter of beads. And we compared this sequential approach with a normal enrichment, so just doing one round with 5 microliter of beads. And we also compared it to the cumulative approach. So here we tested it again with the separate alloys as well with the pooled alloys. And we saw that we can get a significant gain when using this sequential strategy with increasing bead volumes. So this pooled sequential approach was superior to a standard enrichment when starting with input amounts of at least 15 micrograms. So here you can see that the pooled approach is better than the normal as well as the cumulative for 15 and for 30 microgram. However, we didn't observe such an improvement for the 5 microgram peptide input, which might be due to the reason that maybe the bead volumes weren't that optimal for this peptide input amount. So overall, after these evaluations of sequential enrichment, we were able to confirm that sequential enrichment approaches can definitely significantly increase phosphopeptide identifications to a just standard normal one-round enrichment, and also that this pooled approach definitely works and can reduce your MS measurement time as well as avoid these complications with the quantification questions. So as I mentioned in the introduction, the main drivers of advancement in the phosphoproteomic field in the last year have been mainly the technological advancements. And after optimizing our phosphopeptide enrichment workflow, we decided that we want to complete our evaluation by benchmarking this optimized workflow by using the latest generation proteomics mass spec, the Orbitrap Astral. And to do so, we started in our workflow with a HeLa cell dilution series. So we diluted the cells from 1 million to 10,000 million of cells. So by doing so, we wanted to represent typical biological experimental starting points. And then we went on with the protein aggregation capture digestion that is usually employed in our workflow. Then we used our optimized 2-runs pooled sequential enrichment approach, and we measured the samples on the Orbitrap Astral MS. So our results reflected the high sensitivity of the Orbitrap Astral MS. So in the bar chart here, you can see that with the highest peptide input amount, which was 1 million, that corresponds to approximately 59 micrograms of peptide, we were able to identify up to 35,000 well-localized phosphopeptides. And with 500,000 cells that correspond to approximately 34 micrograms of peptide, we were still able to identify 32,000 phosphopeptides. And if you remember from the very initial slides of my presentation, where we used 30 micrograms of peptide as input with subsequent measurement on the Explorer's Astral, we got there roughly 16,000 phosphopeptides. So this represents up to 2-fold increase of phosphopeptide identifications for this input amount. And even with very strongly downscaled cell input amounts, such as 50,000 cells, which correspond to approximately 3 micrograms of peptide, we were still able to obtain a quite comprehensive phosphoproteome with up to 7,000 phosphopeptides. So as these are quite huge numbers, we wanted to see whether these very deep phosphoproteomes also improve the downstream functional analysis. So to do so, we mapped the number of known regulatory sites for transcriptional factors contained in the phosphoproteome of the 1 million input. And we could identify relevant regulatory sites in transcription factors such as CHUM or ATF2, which were distributed across the full abundance range of the obtained phosphoproteome. And then we also evaluated how it looks with the coverage of known signaling pathways. We used here as an example the EGFR signaling pathway, and we were happy to see that we were not only able to obtain information for almost the entire network, but we could also map the most relevant regulatory sites involved in EGFR signaling. And after we had checked the functional downstream analysis with the phosphoproteomics Astral data, we also wanted to have a look at the quantification quality. To do so, we designed an experiment in which we prepared 12 replicates of control samples from HeLa cells. And we also prepared 12 replicates of HeLa cells, which we treated with EGF for 8 minutes. So first, we evaluated the effect of the sample size here. And in this regard, we searched the data from 3, 6, 9, or 12 replicates separately in SpectroNote to avoid a transfer of IDs. And when looking at the average identifications between these replicates per cohort, you can see that the phosphopeptide identifications increase with an increasing number of replicates. But when you apply a quantification threshold of 75% valid values, as it is very often used in typical proteomics experiments, you can see that the depth of the phosphoproteome was similar independently of the replicates used in the search. And following this, we also calculated the CDs here between the replicates. And we were happy to see that independent of the number of replicates, the CVs didn't increase here. So this actually showed us that our full workflow from cell culture over phosphopeptide prep to MS acquisition and analysis is consistent across all replicates, since introducing more replicates here did not increase the CVs. And finally, of course, we also wanted to have a look at the EGF treated samples. So here we evaluated the impact of using more replicates for the comparison control versus EGF. And as expected, of course, when you use more replicates due to the increased statistical power, you will get more significantly differentially regulated phosphosites. But we were happy to see that when we explored relevant EGFR substrates and sites involved in EGFR signaling, that these were consistently upregulated across the majority of the analyzed replicates. Although, of course, you get higher statistical significance if you use a higher number of replicates. So to sum up our findings of these three parts, we saw that during the optimization of the enrichment conditions, mainly the beads to peptide ratio, the binding time, the peptide input itself of the sample, as well as the glycolic acid concentration in the loading buffer can play a significant role on the resulting phosphoproteomes. And we advise users to adjust their parameters for either the highest phosphoproteome coverage, enrichment efficiency, or proportion of multiply phosphorylated peptides. So during the sequential enrichment evaluation, we saw that such approaches can definitely significantly amplify the phosphoproteome depth. And we suggest to use a sequential enrichment approach with pooled alloys with 2 to 3 rounds of sequential enrichment and increasing bead volumes to get this advantage of sequential enrichment without sacrificing MS measurement time and complicating the subsequent data analysis. And this can give up to 20% boosted phosphoproteome depth. And regarding our evaluation on the Orbitrap Astral, we were able to increase the phosphopeptide coverage up to 2 times without the need to scale up our starting cell amounts. And we were happy to see here that with this workflow, we can achieve repeatable deep phosphoproteomes. So thank you a lot for your attention. I'm happy to answer any questions. And also a huge thank you, of course, goes to my own lab, the Ghana and the Maya Matthews group. And also a huge thank you goes to Jesper Olsen and the whole group for this great collaborational work. Also to Anna for the amazing supervision, and also to Ilaria and Claire, who also contributed to this work.
Thank you very much, Patricia. This was great. I think a very thorough and systematic kind of evaluation of different parameters. I think we already have a few questions coming in, so I'm just going to jump straight into them. The first one is from Owen. He's basically asking about the input amounts and whether you've tested anything above 30 micrograms, and specifically doing that with the sequential enrichment, whether there's any benefit to doing sequential enrichment with anything higher than 30 micrograms.
Okay, great question. So for the very initial evaluations, we only went up to 30 microgram because that is the optimal input amount for the whisper gradients on EVO step 1, which are made for the rather low peptide input amounts. But for the sequential enrichment, it would definitely make sense to do this for more than 30 micrograms, because you saw this trend that when you use more peptide, you can also get a gain in the rather later enrichment rounds. So we didn't test it for more than 30 micrograms, but I would expect that you can also get a significant gain with more than 30 micrograms.
Yeah. And I think another consideration maybe to put out there is as we increase the input amount, we also increase our contaminants. And so the front end sample prep in terms of digestion and potentially the salting, which becomes really relevant as you increase the input amount, is going to affect your enrichment efficiency a lot. So I believe the initial data you did was with the desalted sample. And then the subsequent data on Astral was that basically straight from lysate all the way to enrichment without desalting. How was that?
Yes, exactly.
Yeah, exactly.
Yes, it was without desalting because the input amounts were already so low that we didn't want to have any losses here. Yeah.
Exactly. So in fact, you might actually gain by going lower, having lower contaminants and avoiding the desalting. And with a new generation, more sensitive mass specs, I think you just get very good coverage still. So that's one thing. So we have another question about, I think the sequential enrichment, and this is about different bead chemistries. If you've tried, combining different IMAX, titanium dioxide, and whether this will have some benefit of doing the sequential enrichment.
So we didn't test different beads, but since it has been proposed before that different metals can have different affinities to multiply phosphorylated species, for example, I could imagine that you could benefit from it. But within this work, we only tested the zirconium IMAC HP because this is what we found to work best with this workflow.
Yeah. And I think there'll probably be some gains. I think overall, once you start pulling back the eluates, unless you want to analyze them in parallel, you start decreasing those gains. So and it's going to be a balance between mass spec time versus essentially how much depth of coverage you would get. Something that we discussed offline the other days. So this was all done on automated Kingfisher platforms.
Yes, exactly.
Do you have any tips and tricks if you want to do this workflow in a manual format? I know you've done a little bit of work on that. We can maybe share your experiences with that.
Yeah, exactly. So yes, so this we did back in the Olsen lab on the Kingfisher, and then I tried to apply this workflow in our own lab here, and we wanted to do it manually. And what we saw within the first few tries, we had a lot of unspecific binding. So it seems that when you use the Kingfisher, which pulls out the beads from the samples with the magnet, this actually influences the whole workflow. So we observed that when doing it manually, you have to do more intense washing steps in our case. So we had to increase our washing buffer volumes. We had to increase the time for how long we are washing. And also what was quite interesting for us to see was that we had to switch from using a normal thermal shaker to using a overhead rotating thermal shaker, because it seems that the zirconium IMAC beads are quite heavy compared to some other beads that are out there on the market. And you just have to ensure that the beads obviously mix with the sample very well, because otherwise you also won't be able to wash away the non-phosphorylated peptide species in all of the washing steps. And that explains why we had so much unspecific binding. And yeah, we were happy to see that we could solve it with just increasing the intensity of washing. Yeah.
Yes.
So
sorry to interrupt. Carry on.
No, just wanted to say that it will definitely make a difference whether you use it on a Kingfisher or if you do it manually. So you always have to adjust within your workflow.
Yeah, and I have to say the Kingfisher has quite intense washing steps and how it mixes. I think the RPM or the strength of the mixing is set on the highest level for the phosphopeptide enrichment. So it's sometimes tricky to replicate this in manual format. So it's good that you have a workaround in terms of the additional mixing in a larger volume that works quite well. We have another question from Ignacio. Hi, Ignacio. I hope you're doing well. So do you test comparing your optimal enrichment conditions versus the beads recommended conditions in the Astral? See, I'm not sure if I'm understanding this question. So did you test comparing your optimal enrichment conditions versus the beads recommended conditions in the Astral? Ignacio, do you want to answer your question live? I can activate your mic quickly. Maybe you can clarify. Let me see how to do it. Just want to find you on the list here. Nope, I don't see him here. So maybe he's dropped out. Okay, I'm not really making sense of this one, but right. So we can maybe go to the. It says yes, please. So he's here. Why can't I see you? Ah, there we go. Allowed to talk. Here we go. You can, Ignacio, can you?
Thank you.
Hi, Ignacio.
Hi. Thank you, Patricia. No, I meant that if now that since you tested your optimal conditions for phosphopeptide enrichment with the sequential enrichment and proper volume, etc., in the Astral, I wonder if you also had the chance to test the standard conditions recommended for the IMAC beads as well in the Astral, and compare the results and what if there are any differences in the identifications. And I thought that might be interesting since the Astral is such a powerful instrument that reaches like a lot of depth. Did that make it clear?
Yeah. Now I get the question. Thank you for clarifying.
Thank
so
you.
For the Astral here, we only tested the optimized workflow, like the sequential with the increasing beads. It would, of course, I understand that it would make sense to also test the standard workflow, but we just got to the sequential enrichment to work so efficiently and so well that for this project, we didn't see why we should go back to the standard. But I guess that maybe with the Astral being so sensitive, the increase you get with such a sequential enrichment might be slighter, the effect, than what you observed on the Astral, on the Explorers, if you know what I mean. So since the Astral is so sensitive, the gain you get from the sequential versus standard, it might be smaller than on the Explorers, but for this, we didn't test the standard conditions here.
Thank you so much. Thank you, I understand.
Cool. I don't see any more questions. Maybe just another one popped in. Let's see. Yeah, here we go. From Sandra, have you looked at the linearity of the enrichment with a different number of washes? I.e., if you do fewer washes, since you're pulling down less of the most abundant phosphopeptide, is the calibration curve still linear?
Okay, so we didn't check doing less washing steps because on the Kingfisher, the washing steps and how they are made are already so efficient and fast that there is not really a need to scale down on the washing steps. Also, the enrichment is very fast, so not really a need to scale down the washing steps. But definitely when I did the enrichment manually, I definitely saw that when doing less washing steps that we got more unspecific bindings. So I would say that for the manual workflow, it can definitely matter. But for the Kingfisher here, we didn't test scaling down the washing steps.
Yeah, and I think this will be the case also for different types of sample inputs. In this case, you work with cells. I'm not sure if you've started to work with other sample sources like tissues, but these will definitely have an effect on how the enrichment or the enrichment efficiency. And so the front end, again, digest preparation becomes really, really critical in this respect.
Yeah, definitely.
Yeah. So I don't see any more questions. I want to then start closing off and just say that thanks for the insightful presentation, all the details. I think these are now all the conditions that you've tested and are published, so people can have a look at the publication that I think you've got in a QR code there. And
yes, exactly.
Yes, and as I said, we'll have this webinar up on the YouTube channel soon. So if you guys want to rewatch it, you can do so by next week. And I think if you have any questions, you can reach out to us, or I think to Patricia as well. She'll be able to address those. And yeah, thank you very much, Patricia, for your time. And thank you everyone also for your time for joining us today. Look forward forward to seeing you in the next episode.
Thank you for the invitation.
Thank you. Bye-bye.
Bye.
Bye, everyone.